that's a bit of a controversial topic. There are several opinions going. I think probably the original, or one of the first ideas that came up, was that neurons, which is the pool where most or almost most of amyloid beta comes from, some of these neurons become compromised, and then they realize there is a release of intraneuronal contents, including amyloid beta, onto the extracellular media, and that leads to precipitation of those components and the formation of a plaque. Randy Nixon talks about pentos, which is a form of, let's call it poisoned neurons, which have a huge accumulation of undigested autophagic plaques. And these neurons eventually become dysfunctional, and they can actually attract each other, and they end up forming areas with a lot of amyloid beta content, high content of amyloid beta, which comes from this compromised pentos, poisoned neurons, basically having to do with impaired autophagic flux. Then another vision, for example, Gunnar Guras and others think that amyloid beta is released at the synapse, and plaques might come up, precipitation extracellular might come up starting at the synapse. And then, there are others who think that plaques come from the spread of extracellular seeds spread by microglia or other cells. For example, the Green Lab, they were the first who ablated microglia in the brains of Alzheimer's mice, and what they saw was that plaques just disappeared. So that gave us the idea that microglia might play a role in plaque spreading. So I think all these could be valid and might happen simultaneously or different stages of Alzheimer's pathogenesis. What we think is that one of these mechanisms might have to do with microglia. They go engage a neuron that's compromised, which contains amyloid beta fibrils, or engage a plaque, internalize material, go away somewhere else, and release it during digestive exophagy. So we think that could be one possible mechanism that leads to plaque spread.
I agree. Yeah, I agree with what Santi mentioned. Yeah, I think it's all of those are happening simultaneously. It's very hard to just pick one different mechanism where this is happening. But I think when it comes to our project, what makes this a lot more interesting is that the idea of the spread. Because if you think about microglia, they are known to migrate from one region to another, right? So in our mechanism that we're showing that you have extracellular degradation, and at the same time, as Santi mentioned, we do see proof that there is a release of those fibrils, right, that's being deposited on the plaque. Like those are cells that when we were doing the experiment where they were loaded with fibrils, right, and then we add plaques on top of those cells, and we've seen the secretion of those fibrils on those plaques as the cell was trying to digest it. So if we're looking at that in vitro assay, when trying to compare what's going on in vivo, we can, it's not too far-fetched to surmise that maybe this could be a mechanism where you can have spreading of fibrils, at least fibrils, that's being seeded to a different region, and that can form a new plaque, and you have a lot more spreading. So I think this is interesting in that aspect when you look in terms of spreading of plaques in the brain when it comes to Alzheimer's. Yeah. At least one component of it.
And actually, so the MyD88 pathway regulates lysosomal exocytosis, also in microglia. We tested that by treating the cells with inhibitors of downstream cascade with PI3K and AKT1/2 inhibitors. And what we saw was that when we treated the cells with these inhibitors, they were no longer able to release lysosomal contents at the same rates. And then there was a paper that was just published in which the authors treated astrocytes with MyD88 inhibitors in mice, and they saw a rescue in neuropathology. And they actually prevented neuropathology, which indicates that it will be very interesting to see if what happened was that astrocytes, which have also been reported to internalize fibrils, it will be interesting to see whether what happened was that astrocytes also engaged in digestive exophagy, and by inhibiting MyD88, they just stopped releasing lysosomal contents, which in principle might, if we assume that lysosomal release is responsible for spread of plaques, inhibiting that might also stop the spread. So I wonder if that's what happened with astrocytes. But the fact that they inhibited the MyD88 pathway is suggestive that that's likely what happened. So I think that's very encouraging to see on our side.